All right, so in chapter 41, we're going to talk about diabetes mellitus. So we know that for normal metabolism, there's a lot of glucose homeostasis that needs to occur in our bodies. And that homeostasis is mediated in part by hormones like glucagon and insulin. We know the role of glucagon is to raise our blood sugar, and the role of insulin is to decrease our blood sugar. And the way they do this is they act on different sometimes different, but often the same metabolic processes. Okay, so we find then is that if we need energy requirements to be met by glucose and fats, what we can do is actually produce glucose from endogenous glycogen sources. So what's cool is that when our blood sugar is high, our liver and our skeletal muscle can store glucose for later use in the form of glycogen. Glycogen is a polysaccharide. It's a polymer of glucose. It's a long chain of glucose that you store in your liver and skeletal muscle. If your blood glucose is too low, you can start to break down your own body's glycogen through a process called glycogenolysis. Glycogen, right? So glycogen, and then lysis means to break apart. So glycogenolysis means to break down glycogen into glucose. This would help raise your blood sugar back up. It turns out glucagon stimulates glycogenolysis. And that makes sense. That's one of the ways that it raises blood sugar. Now, you can also manufacture glucose. Your liver can actually, remember, one of the functions of the liver is to interconvert nutrient types. So your liver can make its own glucose, but not out of thin air. It has to make its glucose from things like amino acids and lactate through a process called gluconeogenesis. Gluconeogenesis means glucose. Neo means new. Genesis means to create. So gluconeogenesis means to create new glucose. You can do this from protein or amino acids and lactate, which is a breakdown product of other, it's a metabolite of other chemicals, and your liver can make new glucose. So glycogen would stimulate both these processes. Glucagon. Glucagon would stimulate glycogenolysis to raise blood sugar. It can also stimulate gluconeogenesis to raise blood sugar because they both lead to blood sugar rising. Okay. Now, other sources of glucose could be from your gastrointestinal tract, where you can absorb glucose from the food you eat, or your liver, because your liver can actually release extra glucose. Now, we know that insulin is antagonistic to glucagon, and it's a hormone that's synthesized by the beta cells of your pancreas in the islets of Langerhans. And these beta cells produce proinsulin. Proinsulin is converted to insulin, and then insulin is the active form here that goes on to a lot of your body's tissues and stimulates those tissues to uptake glucose. Think of insulin as the key to get the door open to get glucose into the cell. Without insulin, most of your body's cells can't get glucose inside. They need insulin as the key to open up the cellular door to allow glucose to come into your cells. Otherwise, without insulin, or maybe if your body can't respond to insulin very well, then your body's cells can effectively starve because they can't get glucose in. But the way that insulin lowers blood glucose is that it actually helps remove glucose from blood and puts it into your body's cells. That way it lowers blood glucose, right? So you're not actually removing it from your body, you're just removing it from the blood by putting it into cells. That's what insulin's going to do. So in a little bit when we talk about diabetes, we'll talk about how it's characterized where cells have defective transport of glucose into their cell due to either a lack of insulin or the inability to respond to insulin. We'll talk about that. Now we know that the alpha cells produce glucagon, and glucagon antagonizes insulin by stimulating gluconeogenesis, remember, to make new sugar, or glycogenolysis, means to break down glycogen into glucose. Now, our delta cells produce somatostatin, which actually inhibits glucagon and insulin, and then F cells produce pancreatic polypeptide, which actually plays a regulatory role by also inhibiting somatostatin. Now, glucose is the stimulus for insulin release. So it turns out that our beta cells of the pancreas have little glucose receptors, and when glucose levels get high, those beta cells then will release insulin. Okay. Now, how do we know what high is? Like, what is like a high blood sugar level? Well, that's defined by our genetic factors, right? Like, this is something that's built into our biology. And when blood glucose levels get beyond their normal set point or ideal range, then these beta cells then can release insulin. Okay. Now, the effect of insulin is that it inhibits gluconeogenesis. So by inhibiting gluconeogenesis, then you're preventing the liver from making new sugar, right? It also stimulates glycogenesis, which is the process of forming glycogen. Remember, glycogen was a big old chain or polymer of glucose. So if insulin stimulates glycogenesis, what you're doing is you're taking glucose out of the bloodstream and then putting it into long chains of sugar, which we call glycogen. And there's only two places that glycogen is stored in your body. That's the liver and skeletal muscle. It also enhances fat deposition by preventing lipolysis or breakdown. And it stimulates the release of somatomedin, which are these insulin-like growth factors as well. Now, what we see here, you guys, is some of the activities during the fed state. Now, we call the fed state right after you eat a meal, right after you were fed. What's happening here? Well, you're absorbing a lot of glucose along the digestive tract. We can see that here, right? The big G here means glucose. That's getting absorbed into your bloodstream. But where does that go? Well, a lot of that goes to your neurons because 20% of your body's energy, your metabolic activities, are used to control or run your nervous system. So your nervous system is actually pretty costly as an organ system. It only accounts for maybe 1% of your body's weight, but it uses like 20% or more of your body's energy. So a lot of that glucose gets used by your neural tissue. What's interesting though, you guys, is that neural tissue does not require insulin. Your neurons can absorb glucose independent of insulin. So we'll come back to that, why that makes sense. Now, we also know that if blood glucose levels rise, the pancreas releases insulin. And then what insulin can do is enhance the absorption of glucose by the liver, by your skeletal muscle, and adipocytes. Now, if glucose is being absorbed from blood, what's happening to our blood glucose then? It's decreasing, right? And that's the effect of insulin. So what if you had too much insulin release? What happened to your blood glucose levels? Yeah, get too low. And so we know there's a hormone that prevents that. Now, somatostatin, because somatostatin prevented glucagon and insulin from being released too much. And so these processes are normally kind of leveled out and smoothened out. That way you don't get wide fluctuations in blood glucose levels. Now, in the fasting state, glucose is produced in a variety of ways. So where is glycogen stored in our bodies? Liver? Skeletal muscle, right? And it's stored in the form of, actually I said glycogen, that was the question, sorry. What can you break down glycogen into? Glucose, right. So in the fasted state, like you haven't eaten in a while, then you can start to break down glycogen from your liver and your skeletal muscle. That glycogen, when it's broken down through glycogenolysis, that releases glucose in the bloodstream, which raises your blood glucose, right? You can also get glucose in the fasted state through gluconeogenesis, where your liver can make new glucose molecules from amino acids or protein, as well as lactate. So that's pretty cool. And what these can do then is help increase your blood sugar during a fasted state. So hormones that help increase blood sugar could be things like your corticosteroids, epinephrine, growth hormone, as well as glucagon. All of those are anti-insulin hormones, and they act to raise your blood sugar. It makes sense. Remember the slide we saw earlier about the synergistic effects of hormones? When you added those three hormones together at the same time in your bloodstream, you saw the biggest increases in blood glucose levels, right? We had glucagon, epinephrine, and cortisol. Makes sense. So those are all going to increase your blood sugar dramatically. So, but what this is showing you guys is glucose in the fasted state. Notice that our neurons are still using glucose, even if you haven't eaten in a while. So it's kind of interesting that, we understand that we don't have to always be eating to get energy. We understand that you can eat periodically, and your body kind of takes it over from there. You don't have to worry about constantly eating to keep your brain alive and running, right? But we do know that your nervous tissue is always using glucose. In fact, it's kind of like glucose hog. Like I said, it uses 20% of your body's energy just to control 1% of its tissue by weight. So it's a very metabolically costly tissue, but it's the thing that basically makes us us, right? Makes how you think, love, feel, that kind of stuff. So the pancreas releases glucagon when blood sugar is low, and then glucagon can actually stimulate glycogenolysis and gluconeogenesis by the liver. It can also stimulate the release of free fatty acids by adipose tissue, and these can be utilized by other body tissues like muscle tissue. Okay? Now this is supposed to show in the fasted state you're not absorbing any more glucose here. Now what we can do is actually measure someone's blood glucose in the fasted state. Okay? Under normal circumstances, if you haven't eaten in a while, should your blood glucose levels be really high if you haven't eaten a meal in like 12 hours? No. That's not normally, right? Because normally your body cells can use that glucose. But what if you had a deficiency of hormones that allowed your cells to uptake that glucose and use it? Well, then you're going to have elevated blood glucose for a longer period of time. It's going to take you longer to have your blood glucose levels fall, right? So what we can do then is actually measure glucose in the fasted state to figure out someone's glucose tolerance, right? How well do they tolerate this nutrient, and how well can their bodies utilize it? And that can give us an idea of whether they have like pre-diabetes or diabetes. And we'll talk about that in a little bit. So there's different classifications of glucose intolerance. We have type 1 and type 2 diabetes mellitus. Type 1 diabetes we typically talk about as being an autoimmune etiology, although that's still not even totally clear if that's the main etiology. Here in Colorado at the Barbara Davis Center for Childhood Diabetes, they're looking at genetic markers of diabetes. And a long time ago, I was a part of a research study where we were gathering basically DNA from type 1 diabetics and then looking for similar markers between those individuals. And we're getting closer. There are genetic markers for diabetes, but not everyone who has type 1 diabetes necessarily has a family history of it. So there seem to be environmental triggers as well. It's still not clear what those are. But type 1 diabetes, you guys, just to describe as a disease, is insulin deficiency. Okay? So you can't make enough insulin. We know that insulin normally acts to lower your blood sugar, but type 1 diabetes, you don't make enough insulin. So what this means is that they can't tolerate glucose as well, because when they go to ingest some sugar, because they don't make enough insulin, their blood glucose stays higher longer. You might wonder, that doesn't sound so bad, right? Why is it bad to have glucose in your blood? What if I told you guys, though, that glucose in your bloodstream is actually pretty unstable, and it likes to stick to things. It sticks to protein, and it caramelizes those protein. Not like literally in the sense like that we use the cooking word, but it does stick to protein, makes them sticky, and it also initiates immune responses that cause inflammation and damage, so that having glucose elevated in your bloodstream for too long is actually damaging your body. In a lot of ways, you can consider it as a toxin. It's interesting. So type 2 diabetes is actually not insulin deficiency, but rather insulin insensitivity. So with type 2 diabetes, you still make enough insulin, but you're not able to respond to that insulin as well as you could normally, right? So an example of this, you guys, would be someone who has had insulin receptor downregulation. Like their insulin levels have been high for so long that their body cells have now downregulated their insulin receptors, which means they're no longer as sensitive to insulin, so that even with a lot of insulin present, their body cells don't respond to it, and then they don't absorb glucose as well, so they keep having elevated glucose levels for a longer period of time. There are other forms of diabetes, you guys, like gestational diabetes or pre-diabetes. In fact, a pretty large portion of the American population is believed to be pre-diabetic. And the reason for this is that we just have way too much sugar in our diet. I don't know if you guys have ever seen that infographic before, but there's a graphic showing like what the average American child eats in sugar every year. It's about their own body weight. So they showed a 9-year-old child just kind of standing there. Then they had, however heavy a 9-year-old is. What is that, like maybe 60 pounds or so? I don't even know. Anyways, they showed an equivalent weight in sugar right next to them, because that's what the average American child eats, their body weight in sugar every year. So we might think, who cares, right? Just sugar. Why not? Well, the thing about that sugar, you guys, is that if it's elevated too long, first of all, it can damage your body's tissues, and it can lead to things like type 2 diabetes. We used to call type 2 diabetes, by the way, adult onset diabetes, because it used to only occur in adults. Now it's so common in children, we can't even call it adult diabetes anymore. It's called type 2. And that wasn't that long ago, guys. It was only 30 years ago. So, interesting enough. But some different lab values here, you guys. Impaired glucose tolerance, you'd find 140 to 200 milligrams per deciliter 2 hours after glucose. Or impaired fasting glucose tolerance would be sort of fasting glucose values in the 100 to 125 milligrams per deciliter range. Now with type 1 diabetes, it's characterized as a deficiency of insulin. So the beta cells don't release insulin. It's believed to be an autoimmune etiology where someone's immune system is actually killing off or attacking their beta cells. And because their beta cells are being removed, then they actually have deficiencies in insulin in the long run. and it's diagnosed usually between 5 and 20 years of age. Type 1 diabetes is usually the one we talk about is running in families and being more genetically linked. In fact, type 1 diabetes is more common in individuals of European descent. Okay? I don't mean necessarily like in the last 3 or 4 generations, but I mean like European descent within the last maybe 200 generations. Okay, so maybe like in the last 300 years or so. I'm not actually, no, that's not right. I meant 1,000 years. So people of European descent. But you know, if you have ancestors that were Europeans, then that can also put you at high risk of type 1 diabetes. So don't just think about like white people, because there's a lot of intermixing, you know, throughout history. So think about people of European descent, maybe you know, breeding with other people around the world. Then you can see higher rates of type 1 diabetes in those parts of the world. Like you also see it in North Africa. You know, you think, oh, well, North Africa is Africa, right? Well, North Africa used to be part of the Roman Empire, and well, those are Europeans, and so you find higher rates of type 1 diabetes in North Africa as well. So it's kind of interesting. So its etiology may be immune related. There seem to be some genetic markers like this human leukocyte antigen DR3 or DR4 seems to be a risk factor for type 1 diabetes. And there's a really interesting hypothesis about why this exists. We know that animals can survive winter and when it freezes, right? Like how is it that frogs in a pond can just emerge the next spring and be like ready to hop around and be a frog and like ribbit and eat flies and stuff? Where do they go all winter? What if I told you guys they froze? They didn't freeze to death though, because before before the freeze came, those frogs started pumping their bodies full of sugar, and they started excreting a lot of water. Well, sugar is an antifreeze. In fact, if you have really sugary drinks with not a lot of water, it has a lower freezing point. So these frogs can freeze solid in the winter, but their cells don't burst because they don't have a lot of water. We know that water expands when it freezes, right? So the hypothesis is that individuals with type 1 diabetes may have been better adapted to surviving extremely cold environments, where if you were type 1 diabetic and your blood sugar levels are really high, then you're more likely to survive a deep freeze because now your cells aren't bursting. It's a hypothesis, but it makes sense with respect to more recent ice ages, you know, the last 1,000 years or so. So it's kind of interesting. Now, it results from absolute insulin deficiency, and this overproduction of glucagon can stimulate glycogenolysis and gluconeogenesis. Now, as glucose levels rise, this can lead to polyuria. The reason why you get polyuria, you guys, is this causes glycosuria. Glycosuria is sugary urine. Sugar is a solute. If you have such high blood sugar that your kidneys can't even reabsorb it, then you're going to lose some sugar in urine. Well, sugar is a solute. Water follows solutes, so it's going to increase urine output. So one of the signs of diabetes is polyuria, which comes from sugary urine. In fact, one of the ways they used to test diabetes back in the day is they'd actually test to see if your urine was sugary, either by tasting it, right? They taste to see if it's sweet. If you had sweet urine, they say, oh, you got glycosuria, that's diabetes, right? Although that's kind of a gross, that's not very hygienic, and kind of a gross way to test for diabetes. You know what they did in traditional Chinese medicine is they looked to see if ants were attracted to your urine. Makes sense, right? Normally ants shouldn't be like, ooh, hey, urine, right? But if there's a lot of sugar in it, yeah, they'll be attracted to your urine. And that's one of the ways they can test for diabetes back in, you know, China 3,000 years ago. It's actually kind of cool. Now, as these glucose levels rise, it causes polyuria. Now, with that polyuria, you get polydipsia because you get so much water lost from excessive urination that you want to recuperate that volume by drinking a lot. Okay, and you get polyphagia because what does insulin do to most of your body's cells? It lets glucose in. If you have type 1 diabetes and you don't have enough insulin, the glucose is there, it just can't get in. So what happens is their cells start releasing hormones saying, hey, I'm hungry, right? Because the cells are starving. They're not getting glucose in the cell. So they release those hormones saying, hey, I'm hungry, like ghrelin, that kind of stuff. And that stimulates you to be more hungry, which is what we have this here, this polyphagia. Phagia means to eat, right? Polyphagia means excessive hunger. So these are the cardinal signs of diabetes: polyuria, polydipsia, polyphagia, and glycosuria, or sugary urine. Sweet urine. Shouldn't normally have sugar in your urine, because that's a nutrient, and you want to reabsorb that. but you can overload your kidney's ability to reabsorb sugar. So free fatty acids are converted to ketones as a way to keep your nervous system running, because under really severe circumstances, your brain can run on ketone bodies rather than glucose. Remember I said to you guys that the nervous system only relies on glucose? It's true for the most part, but under really severe circumstances, it can run on ketones. These ketones have a sweet smell to them. In fact, they can dissipate through your skin and mucous membranes. So people that are undergoing ketoacidosis have a sweet or fruity smell about them. And you know that type 1 diabetics in ketoacidosis because you can smell it. I mean, I've smelled it. I wish I could describe it in a better way other than sweet, fruity odor. But if you guys know somebody with diabetes and you smell that they smell differently, like kind of sweetish, not Swedish, but sweetish, then that's ketoacidosis. Those are ketone bodies. Now, type 2 diabetes is where you have insulin insensitivity. Okay, so we can still make insulin, but we're just insensitive to insulin. Our body cells are like, the insulin's there, they're ignoring it. Remember I gave you guys the example of like tissue resistance as those unruly children who just stop responding to like any kind of parental scorn, right? I'm thinking of my stepsister. Like, she gets yelled at so much, it doesn't even faze her. But I remember when I was a kid, if I got yelled at, I would take it so to heart that like it would actually, maybe do something and maybe I wouldn't be so bad, right? But with her, she'd just get yelled at. No problem, she doesn't care. She's so used to getting yelled at, she doesn't even respond to it anymore, right? So, probably shouldn't go into any more detail than that. But what about your body's tissues? What if you're around insulin so much that your body's cells just stop responding to insulin? There you go. That's tissue resistance. So tissue resistance comes from downregulation. And that downregulation is just basically your insulin levels are so elevated for so long that your body's natural tendency then is to downregulate receptors that we don't have such a strong response to that insulin. So this insulin resistance can lead to basically high blood sugar, even though insulin's present, right? You can have insulin present, but you still have high blood sugar because your cells aren't responding to that insulin. Okay. Now we typically talk about type 2 diabetes as the one that's acquired in life, right? In fact, what kind of diet would stimulate you to have a lot of insulin being released over a long period of time? Yeah, high sugar, right? High in carbohydrates. Carbohydrates are sugars. There's simple and complex carbohydrates. Simple carbohydrate would be like table sugar, right? Complex carbohydrate would be something like starch, like in a potato or bread, right? Those are more complex carbohydrates. And your body treats them like sugar. It is sugar. Basically, you can eat a potato, and it has the same sugar content as a certain amount of table sugar. Now, although your body does react differently versus a potato and table sugar, I mean, sure, we can all estimate that. Like, you wouldn't eat a handful of table sugar and say, oh, that's nutritionally equivalent to a potato, right? Because potatoes have like fiber and stuff like that, so you get slower release of that sugar into your bloodstream. So you have a high sugar diet, so your blood sugar levels are elevated. What does your body do next when your blood sugar levels start to spike? You start to release insulin, right? And as you release insulin, then blood sugar levels decrease. Well, multiply that times thousands of meals. Blood sugar levels spike. You release insulin. But what if your cells have now started to downregulate their receptors for insulin, and they no longer respond to that insulin? So your blood sugar levels spike, then your insulin levels spike, but your blood sugar levels don't change that well, right? We call that glucose intolerance, because even though you're releasing insulin, your blood glucose isn't falling because your cells are no longer sensitive to that insulin. They can't respond to it. So it's there, it's just they're not dealing with it, not responding to it. So that keeps your blood sugar levels high. Now, with type 2 diabetes, though, this can lead to type 1, because as the disease progresses, insulin production could be impaired. So it's kind of interesting. Now, some of the risk factors for type 2 diabetes would be like female sex, obesity, aging, and sedentary lifestyle. In fact, one of the major risk factors for type 2 diabetes, you guys, is just age. As you get older, your risk for diabetes increases. There are some protective measures against type 2 diabetes, though. It's opposite of these. Okay? Obviously, there are modifiable and non-modifiable risk factors, right? You can't modify or change your genotype. Okay, but nor can you change your age. But you can change your body weight and how active you are. So if the idea being that if you exercise more, that actually increases insulin sensitivity. And there's really good evidence of this, you guys. If you just like walk 3,000 steps a day, that itself increases insulin sensitivity. And if you're more sensitive to that insulin, then that's protective against type 2 diabetes. Obesity correlates with type 2 diabetes, but what's not clear yet is obesity a causal factor or is it a correlational factor with the degrees progression? That's not totally clear yet. And everyone thinks, if you're obese, then that's a risk factor for type 2 diabetes. But what's not clear is that did those co-occur, or did diabetes cause obesity, or did the obesity cause diabetes? That's not totally clear yet. So we're still trying to figure that out, but it's interesting. Now, like type 1, you still see polyuria, polydipsia, polyphagia. Why do we have polyuria with diabetes? Water follows solutes. What's our solute? Sugar in the urine. And if you got sugary urine, we call that glycosuria. Good, sweet urine. And then why do we get polydipsia, or excessive thirst? Yeah, because you lost so much water that you want to drink, right? And you got polyphagia because your cells are starving. How are your cells starving? You got high blood sugar. Glucose can't get in. You got it. So ketoacidosis is uncommon with type 2 diabetes, but we can get non-ketogenic, hyperglycemic, hyperosmolar coma. Woo, that's weird. Okay, let's go through that. Non-ketogenic, hyperglycemic, hyperosmolar. This is where it's not due to an increase in ketones, but because your blood sugar is so high, it changes the osmolarity of your blood. And we know that water follows solutes. Well, what if you started to absorb some of the fluid from your brain into your bloodstream? Then your brain starts to shrink. Well, that can lead to coma and possibly even death. It's kind of interesting. Now, gestational diabetes is actually kind of interesting to you guys. And I'll try to explain it from a standpoint of evolution because I think it makes sense in this regard. If you're a pregnant mother trying to feed a fetus, you want to keep your blood nutrient levels high, right? You don't want to have your blood nutrient levels really low because then your fetus would also starve. So you should expect then under normal circumstances that as a pregnant mother, then you want to have blood sugar levels high enough to feed yourself and the fetus. But imagine that normal process amplified. So where you have too high blood sugar, like even more than what the fetus would need, right? That's what we call gestational diabetes, okay? Now, it closely resembles type 2 diabetes, and it's most likely precipitated by placental hormones and weight gain. It's believed that the placenta can release hormones that stimulate the increase in blood sugar in the pregnant mother. It's so weird, you guys, but basically you're saying that the fetus here is releasing hormones into the mother's bloodstream that causes the mother to release excessive amounts of glucose. Yeah. This can lead to weight gain. Some of the risk factors for gestational diabetes, though, could be things like obesity, history of that, previous offspring that are heavier than 9 pounds at birth. So, what's the word for that if it's a heavy baby? No, there's a word for it. Not just fat baby. I forgot. Presence of glycosuria or strong family history of type 2 diabetes. Now, pre-diabetes is impaired glucose tolerance and impaired fasting glucose tolerance. Now, the reason why it's pre-diabetes, you guys, is you don't see as many of the hallmark symptoms. You might not see as much of the glycosuria or the polyuria, polydipsia, polyphagia that you would see with like a true form of diabetes. But it's suspected that up to a third of adults in the U.S. are pre-diabetic, maybe even more. And so it's sort of an intermediate stage between normal glucose metabolism and diabetes, and it's absolutely a risk factor for diabetes and the associated cardiovascular disease. Now, you might wonder, we probably all understand that low blood glucose is harmful, right? Obviously, our brain needs glucose to continually run. Our nervous system needs glucose to function. But why is hyperglycemia bad? Like, why is having high blood glucose a bad thing? Well, we know that glucose is a solute. We just talked about how that can actually change the osmolarity of your blood, and you can start to change your fluid balance, especially like in brain tissue, leading to coma. But we also know that glucose is an unstable molecule. I know it sounds kind of weird, you guys, but if you could somehow measure a bag of table sugar over the course of several years and you just left it alone in your cabinet, that bag of table sugar would get lighter over the course of those years because glucose spontaneously breaks apart into water, oxygen, and carbon dioxide. Now, obviously those things can evaporate and go away from the bag, right? In our body, the glucose can actually split apart into a chain, because glucose is normally a ring. It can split apart into a chain, and that chain can stick onto proteins. We call it glycosylating proteins. I liken this to caramelizing proteins, right? Now, you might know, okay, who cares, right? Your immune system can just remove those proteins, and so what? Well, if you have high blood sugar chronically, you're talking about glycosylating or caramelizing proteins so severely, it's actually causing tissue damage. Some of this can be irreversible. And now normally we should see blood glucose levels kind of rise and fall. We understand that in the morning, glucose levels rise as your body's waking up physiologically. And in the evening, glucose levels fall as your body's falling asleep physiologically. But with hyperglycemia, we talk about some of the symptoms and signs. Can be things like polyuria, polydipsia, polyphagia. Fatigue and blurred vision and nausea from some neurological symptoms that can occur. You're also more prone to infection with hyperglycemia. Sounds weird, you guys, but glucose is a nutrient that many bacteria can use to thrive. So if your blood glucose levels are elevated for a long period of time, then there's just more nutrients for bacteria to live on in your body. So that people with hyperglycemia are also more prone to infection. On top of the fact that hyperglycemia causes micro and macrovascular damage, and if you damage those blood vessels, then it can also inhibit your immune response and proper inflammatory response. So chronic hyperglycemia can lead to systemic changes, can put you at higher risk for things like metabolic syndrome, hypertension, cardiovascular disease, and stroke. And these complications can be vascular or neuropathic. These can often go hand in hand. Now, the macrovascular complications of diabetes are due to, remember, hyperglycemia, so high blood sugar. That sugar, what happens when it breaks apart? It can stick. Glycosylate protein, not just hemoglobin in blood. You guys know, like hemoglobin HbA1c. It's a glycosylated hemoglobin marker for hyperglycemia. But it also glycosylates other proteins in your body, not necessarily the ones just in your blood. What about the proteins that form your vessel wall, right? Remember, like we had tunica intima, media externa? Well, it's supported by extracellular proteins as well. So that sugar can start to stick onto the proteins you find in your blood vessels that are large and small. And it's going to lead to complications like stroke, ischemic heart disease, and damage to peripheral vasculature. Microvascular damage can lead to things like retinopathy and nephropathy, so damage of your retina and kidneys, as well as abnormal thickening of the basement membrane and capillaries throughout your body, which can lead to deficiencies in nutrient exchange, and that can lead to blindness and renal failure. So who would have thought that having high blood sugar can make you blind? But the reason why it makes you blind is that it damages tissues, including your retina. It can also damage your kidneys, too. So it's kind of interesting. Now, in terms of the treatment education for diabetes, you guys, a lot of the goals, especially depending on the type, right? Which one is autoimmune? Type 1, most likely, right? Which one's more lifestyle mediated? Type 2. Now, there's challenges in education in both groups, right? The challenge in education for type 1 is helping to teach someone about their disease and understanding what they need to do to maintain their own homeostasis, right? The challenge for type 2 is also helping to teach someone about their disease and also helping to figure out what they need to do to prevent themselves from having high blood sugar or potentially even reversing their type 2 diabetes. So what's really interesting, you guys, is that especially with type 2 diabetes, treatment is aimed at educating the patient, but also making sure that they're exercising and eating well. And when this says diet, we just mean like a diet that's low in sugar, if that makes sense. You know, having like a low glycemic kind of diet. And hygiene practices too, you guys, because of the vascular damage that can occur with diabetes, they're more prone to infection. You guys heard of like diabetic ulcers and stuff like that, right? Like it occurs in the feet and other extremities. Now these ulcers come about because of damage from those blood vessels. But we know that ulcers are more prone to form if someone has poor hygiene or if, they have a lot of swelling from edema. So maintaining proper hygiene practices can actually help save someone's limb. In fact, you see individuals who are like type 2 diabetic, it can be reversed with lifestyle changes for the most part. And as long as you treat as long as the patient can take care of themselves and they're well taken care of by their clinicians, then they shouldn't have to lose a limb. But what's unfortunate is you see these individuals who like end up losing both their legs from necrosis and that kind of stuff. So for type 1 diabetics, you guys, they need insulin, right? Because type 1 diabetes is insulin deficiency from a lack of beta cells. So 100% of type 1 diabetics need insulin, and it replaces that deficient hormone. Up to a third or so of type 2 diabetics may need insulin. But the type of type 2 diabetics that need insulin are the ones where they can't control their own sugar intake as well. And when their blood sugar gets really high, they need to inject themselves with even more insulin just to get their cells to absorb that sugar. And so for a third of type 2 diabetics, they also need insulin. So it's not true that type 2 diabetics don't need insulin. That's under more severe circumstances. Okay? Now, this replaces the deficient hormone. There's different types of insulin. We'll do a case study on this later, you guys. We'll talk about rapid acting, short acting, intermediate acting, and long acting. And depending on the way this person lives or what time of day it is, they're going to need a different type of insulin, right? So we'll talk about that. And there's different ways to administer the insulin. It can be administered through injections, which aren't fun. You're talking about someone taking a hypodermic needle and having to stick it in themselves maybe multiple times a day, right? Different pumps, which may need to be surgically implanted, or jets or pens. And then we'll talk about the long and rapid-acting forms of insulin later. Now, some of the complications of hypoglycemia. Now, we've talked a lot about hyperglycemia complications. Hypoglycemic complications can lead to a lot of really severe problems, you guys. Like the neuroglycopenic symptoms are going to be the things that affect your nervous system. So it's going to lead to hunger, visual disturbance, weakness, paresthesias, confusion, agitation, coma, death. They even talk about belligerence, right? So they're sort of they get a little more violent, and I guess one way to put it is maybe a little bit more animalistic. Now, it's caused by insufficient food intake, unplanned activity. You know, like if someone especially kids aren't good with this, but let's say if you have a kid who's type 1 diabetic, and it's time for recess, and they go out and they start exercising, but they haven't eaten yet, right? Well, that exercise will cause their blood sugar to fall too low, and then they can start to go into hypoglycemia from that. So, you know, if you're like a type 1 diabetic and they're going to exercise, you got to be really careful about planning their intake of sugar around that exercise, especially before, right? Like before exercising, maybe like drinking a juice box or something before going to exercise. That way they don't get hypoglycemia from the exercise itself. Or inappropriate insulin administration. You guys hear about people like overdosing on insulin. They might do their calculation wrong and they end up injecting too much insulin in their body, and then their blood sugar just falls too low. But remember, if your blood sugar is low, your brain starts to shut down. So they get visual disturbance, weakness, agitation, maybe even coma and death. Now, this can also lead to pallor, tremor, sweating, palpitations, and anxiety. And some people can have hypoglycemic unawareness, where they're so used to having low blood sugar that just feels like it's normal. Like they don't know that they have low blood sugar because they're so used to having it that low. Yeah.
At Denver Health, they have an eating disorder unit.
Yeah.
And a lot of these girls, so they haven't eaten in months. And
Yeah.
they come in finally. So their body doesn't know what to do with the sugars that they're finally getting. So they just have really low blood sugars. And their blood sugars will be down in the 30s.
Yeah.
And they're fine.
They're functioning.
Around,
Yeah.
They don't feel sick or anything.
Yeah, if there were any of us with blood sugar in the 30s, we would be passed out or something. Yeah, it's interesting. Yeah, you're right. So that your body can kind of adapt to low blood sugar. It makes sense from a survival standpoint. Now, other complications of insulin therapy, you guys can get lipoatrophy or lipohypertrophy. This atrophy and hypertrophy can come about if someone's using the same injection site. Like let's say if they're doing injectables and they're injecting insulin in the same exact spot. Well, because the needle starts sticking in the same little area, maybe on their skin, that can lead to lipoatrophy, the loss of adipose there, which can cause their skin to form a divot in that injection site, which, depending on the person, they might not want. This is kind of a cosmetic, thing. Or lipohypertrophy, again, for the same injection site, insulin can cause adipose to grow in a particular area, so you might find they get kind of like a lump in that place that they're consistently injecting insulin. So for someone who's doing injectables, they need to be able to vary their injection site. That way they avoid lipoatrophy or hypertrophy. With excessive insulin use, you guys, you can also get insulin edema because insulin is a solute. It's a protein hormone. It's also a solute. It can attract water. It can start to have you hold on to water in your tissues, and that can lead to edema around your body. And if someone's using a lot of insulin, that can also lead to insulin resistance. Could you guys imagine a circumstance where someone's type 1 diabetic, they have to take exogenous insulin, but they have to use insulin so frequently that they end up getting insulin insensitivity, that it progresses to insensitivity? What do they have left to do then? You're type 1 diabetic, you can't make your own insulin, and then you have to use insulin so frequently because your diet's too poor and you have to keep your blood sugar down, that you start to get resistance to that insulin. What's left?